chemistries with different read lengths. We have only talked about Illumina chemistry at present. I will go in brief on the other chemistries as well. So, we spoke about paired end and mate pair of Illumina. So, in Illumina we have 3 different instruments, that is HiSeq, MiSeq, NextSeq. We also have HiSeq X series which has come, but the chemistry is same as HiSeq 4000. So, we can generate read length of these. So, these are read length. When I talk about read length, what I mean is this particular read will be 150, this particular read will be 150. That is why it is 1 and 2, that is why 2 into 150, right. So, that is what is the calculation of 2 into 150. So, 2 into 150, it can also generate 2 into 100. MiSeq is longer read length, that particular run fetches you lesser amount of data, but gives you a longer read length. So, if I have longer read length also, I can resolve gaps, right. How? So, if I have instead of 250, if I have here 300, if I have 300, I can even better resolve my gaps, right. So, this is how 2 into 300. Now, NextSeq 500 is again an Illumina platform which can generate 2 into 150. These are instrument outputs. So, HiSeq is a huge data output with a somewhere around 1500 GB in one run. It has 7 plus lanes and gives you 1500 GB in one run. So, just imagine if I am sequencing a pathogen, I will just require 4, 5, max 10 GB. How many samples will go in this particular run? So, that small basis of 8 stretch will segregate those samples. So, can accommodate so many samples together. Now, in MiSeq we have only 15 GB of output, but gives me a longer read length. NextSeq again 120 GB. Now, comes the longer read length. In longer read length we have at present PacBio and Oxford Nanopore. Nanopore is upcoming, so it has a maximum read length of 900 KB. Earlier what used to happen is these shorter sequencing chemistry, when we are doing a contig, we get a size in KBs, right. 1 contig or 2 contigs will be in the KBs, right. In this we get the sequencing itself in KBs. Just imagine the sequencing, if it is that big, the assembly becomes even more easier, right. We can resolve many more gaps, but these chemistries have very high rate of error. Why? Because they are introducing indels or introduces errors. So, errors means SNPs, indels. So, we cannot segregate from our original things, it will introduce itself. So, what is recommended is that if you are going for a higher genomes, clubs shorter read length plus longer ones. What will happen? The error rate which is there in this part will get resolved with this chemistry. Each chemistry has its drawback, each chemistry has its advantages. So, advantages and drawbacks, if you want to combine it together and you want to get a good result, we usually combine the different libraries and different platforms, that is known as hybrid. So, we do a hybrid assembly. So, this hybrid assembly helps us overcome the drawback of the other platforms. So, in this we have maximum 15 KB and gives us a data of 5 GB within one SMRT cell, right. So, we have small SMRT cells, in that PacBio is getting sequenced, and in that we get 5 GB in one particular SMRT. In this Oxford Nanopore we have up to 5 GB data of 900 KB, but usually we only end up with 3 to 4 because it is just coming up. We also have a new chemistry coming up in this mini ion. New chemistry has come up which has improved in sense with the error rate, but the error rate is somewhere around 10%. That is a huge number, whereas the error rates in these are 0.08% only. So, that what is the difference in the error rate of these chemistries, right. So, we combine these chemistries together for doing a sequencing. We also have a platform called ion proton or ion S5, which is a thermo based platform, which also gives you around 15 GB in one run, goes up to 200 base pair, but this is mainly for very small genomes. So, if you are doing bacteria or something, and if you have the setup, you can go for these platform as well. But main players in the market are Illumina and PacBio. Yeah. So, when we are calculating, for example, if I want to go for a genome of 50 MB, right? How do we go for a genome? We consider a coverage. I do not sequence 50 MB only. What we do, we sequence 50 into 100, that is one particular region of the genome I want it to be sequenced 100 times. When we talk about coverages, if we have read genomic papers, we have a word called sequencing coverage. We have sequenced it at so and so coverage. If I say 100, the sequencing is done at 100x coverage, that means I am sequencing a particular region 100 times, right. So, this 50 into 100 becomes my 5 GB, right. So, if it is 5 GB, that means 5 GB data I am generating for the genome of 50 MB. And if it is like 100 times, theoretically I am sequencing one particular DNA template, right. It will not be identical. If it is identical, it is PCR artifacts and it will be thrown out. So, it will not be identical, but that particular region of the DNA will be sequenced 100 times. That what is the theoretical meaning of coverage. So, that is how we calculate how a genome has to be sequenced, and that is when you can just imagine how many samples will go in particular run. If I am only generating 5 GB, this is what is the run output of particular platforms, right. So, once we have done the sequencing and we have the data looks like this. So, we have one R1 which I said as forward read, and we have one R2 which is a reverse read in case of paired end. So, how we identify whether it is R1 or R2? It is usually written in the file name of the machine generated file, but still if you have misnamed, we can identify using this. This is one and the same thing is here, and this is the header of the read. This header of the read will have your instrument ID followed by with the flow cell. So, when we are doing a sequencing, we have a flow cell which is barcoded, and universally in Illumina product that will only be unique, right. So, using that barcode, the sequence has this, right. For example, as run fails off, the Illumina can detect which flow cell it is, which lot number it is, and whether that particular lot has gone off, right. So, that is the way you can trace your flow cell, and followed by the tile number. So, each flow cell has its tiling. You have an image system to capture the sequencing. The clusters which are formed with bridge amplification, with the intensity they capture the color. So, which tiling it is, which position, all this information is written in the header. And the important part is this 2 and this 1 and this is 2. The entire header will be same for these two sequences, only difference will be 1 and 2. And this is how a software, any assembler will identify this is my forward read or this is my reverse read, based on which library you are using. Second thing is that the barcode which is written, the 6 base pair barcode is written here, right. So, this you can see it is identical for all, because I have taken only from one file. So, a sample will have identical barcodes, and you can see the R2 also having the same barcodes, right. So, based on this we can segregate the sample wise samples from a run. So, if we have 10 samples in one run, I can segregate 10 files, 10 into 2, because R1 and R2 based on these barcodes. And this is my header line, this is my sequence, ATGC sequence, this is the strand, and this is my quality of that ATGC. At what confidence is this base called? Is this quality? This is an ASCII quality of it. This is how a FASTQ file looks, and this is what is the raw data which comes out from an NGS machine. Yeah?
What is that AAEE are standing for in the quality?
Ma'am, they are basis quality, ASCII score. So, if you convert it, capital A will be 65, something like that. So, ASCII score it is. So, you see the conversion in ASCII, so you can convert it. They are all values. Yeah. Then once we have done a sequencing, we do a QC of the reads. How do we do the QC? We do the QC based on this and this. This is my quality. So, I do not want any poor quality reads to be there. How we can identify that? Based on these graphs. So, if I have dropping quality to 2, which will be an average quality, so I do not want. If I have a read like this, I filter it out, I remove the low quality reads, I remove the reads which are having too many n's, more than 5% n's. That means that read could not identify that particular base. If it is not able to identify that particular base, it will throw off as n's. So, I do not want such reads which already has ambiguity, which will not help me in my assembly. So, we will remove those reads. We will trim off the initial reads. If you see the initial reads, they might have low quality, right. So, we trim off the initial reads based on the quality. If my average quality is not more than QV30 or QV25, I trim off those reads, right. QV35 is a standard quality reads. So, we trim off those reads which are not in on average 30, QV30. So, we trim off these reads, we see overall coverage of average coverage of this. If it is coming below 30, we will remove those reads. If we do that, this particular reads look like this. So, now my average goes up to somewhere around 25 and my median is somewhere around more than 30, right. So, such type of reads we go for analysis. These type of reads which machine throws, we do not, because this can also happen because of the library preparation, it might happen because of the sample condition, the DNA is not properly isolated or the DNA was not of good quality. Many reasons can happen for such type of reads, and we filter it out and we get such reads. Once we get such reads, we go for de novo assembly or reference based assembly. We have two types of assembly, wherein if we have a genome already published, I already have a reference, then I can straight away go and sequence this read and map it on the genome. That is known as reference base, reference, and on that basis I am calling my genome, right. If I do not have a reference at all, I go for de novo assembly. De novo assembly wherein I do not have any reference, I am going to assemble using some assembler, right. So, we will see how it has been done. In layman language, de novo assembly looks like this. If I have a cut papers, it will look like this and I need to assemble it like this. This is what is de novo, wherein I have no clue, based on only overlaps I am going to get to the genome. So, this is how we do a de novo assembly. There are lot many steps for validating the genome assembly and get to a proper draft assembly. So, the raw reads we already saw, that FASTQ reads which we have R1 and R2, these are my raw reads. Then we quality filter, which we see that the quality should not be low, n should not be there in the reads. So, we quality filter the reads and then we go for a primary assembly. Assemblies are again of various types, we will see how they are being done, and we usually use paired end and mate pair in the assembly, and we can also do a hybrid assembly using PacBio or any long read sequencing like nanopore. So, if we are using PacBio, we have a different filtration step, because PacBio reads are longer and they have different sorts of insertion deletions. Now, SQL platform, it filters the data and gives you using smart SMRT analyzer, which is already there in the system. So, it filters the reads based on the quality and gives you. So, all the bases which are there in the PacBio read will be of same quality, right. So, you do not have to filter it based on quality. Now, what we do? We use the short reads and map it on the PacBio data using softwares and correct the reads of PacBio. The reads which are having more number of indels or reads which are having more number of insertion, we see any ambiguity, we remove those reads from my data, and then we take all those data together and do a hybrid assembly. Yeah. In hybrid assembly again we have several stages. First is contig. If I am only using paired end data, I will first do a contig, then I go for scaffolds. Contigs are those which are small stretches of sequences which does not have ends in between, major chunks of ends. Scaffolds are those which connects those contigs together with stretches of ends in it, right. So, we go for contigs and then we go for scaffolds. After the scaffolds, we have lots of stretches of ends. So, we do a gap filling. The gap filling is where the longer insert length comes in play and longer read length comes in play. So, we use the mate pair, we use the PacBio for my gap filling, right. Once I am done with the gap filling, we have a draft genome which is ready for validation. I have a draft genome with gap filled, with contigs, super scaffolds and gap filled genome. So, this is what makes it a draft genome. Once I have the draft genome, what we check is the whether, for example, I am assembling a 50 MB genome, I should get an assembly nearby 50 MB. I should not get an assembly of 1 MB in that. If I am getting a genome of 1 MB, something is very much wrong in it. So, I should get something close to 50 MB with a lesser number of contigs, lesser number of scaffolds. The lesser it is, it will be more towards the original chromosomal level, right. Usually, we will get in thousands, right. The more lesser it is, more accurate it becomes. So, that is where we validate the genome, and then we can do several analysis like functional annotation, comparative analysis, we can go for structural analysis and all. That is like how we can identify the genes from the genome, we can compare it with other existing genomes, several analysis we can do, but this is the base to get to the genome. So, as I said that in this assembly we have several steps. We have several algorithms with which we can assemble the genome. One of the algorithms is overlap layout consensus, that is OLC. So, many of the softwares work on this basis, overlap. How it works? It works based on overlaps. If anybody remembers the graph theory, nodes and edges. So, nodes and edges, these are all mathematical algorithms. So, behind the software this what works, but on when we are using it, we do not actually go to this depth, we are just explaining how it has been done. So, for example, we use overlapping, that is just the sequence matching has been compared. So, anywhere it gets an overlap of certain bases that has been specified when we are doing an overlapping based algorithm, we specify I want 100 bases to be overlapped, I want 50 bases to be overlapped, I want 5 bases to be overlapped. Based on my requirement, I will tell that this much base overlap I require. So, what it does is that this is the way it will find the overlaps between the sequences. If I have raw reads, the raw reads will be getting an overlap like this. So, what happens is nodes are my reads and edges are my overlaps. So, just imagine the mathematical network of nodes and edges, one line here, the circle here, again one circle, one line. So, this network is getting created in millions of reads. Just imagine the computational power required for doing millions of sequencing. When we talk about 5 GB, it is some millions of sequences, right. So, we have overlaps. Each and every sequence finds an overlap with that first sequence, and that overlap is getting built up. And just imagine the capacity which is required to find these overlaps. And once this overlap is found, we have a consensus sequence, this is how a contigs is formed. This is one of the methods. The another method is de Bruijn graph. Most of your assemblers will be working on this algorithm. In this, we the raw read is been assigned a k-mer length. K-mer is nothing but a small sequence length. For example, if I say k-mer, k-mers are usually in odd numbers, right? Cannot be in even number, will always be in odd numbers. 27, 29, 35, something like that, yeah, cannot be even. So, if I say 27, my 150 bases which I am talking about 2 into 150, that 150 bases is chopped in 27, right. The entire, just imagine millions of reads, 10 million reads getting chopped in 27. So many reads, fractions get created. So, these are the small fraction of reads which are getting created. Now, in this, one sequence will be finding its overlap against the other sequence. Each and every sequence will get matched to the each and every sequence of other. Just imagine how many. So, this way the de Bruijn graph is created, and based on this the overlap of sequences gets attached to each other, and when that happens it creates a contig, right. So, this is how contigs are created using several softwares, such as we will see what are the softwares. SOAP de novo is one, CLC is one. These are different softwares. We also have a list, so you can see the list. So, this is just a presentation how we create contig. So, this is a DNA template. We have a forward and reverse reads. This is what I was talking about, the distance between the two reads is called as insert. You can see the red part, this is what is the insert between the sequence. And these are the million reads, how they go and assemble with each other, and they create a contig. Once the contig is created, they create scaffolds. How they create a scaffold is in this way. What happens is I hope I am not blocking you. Yeah. What happens is insert the long contigs into scaffolds. So, what happens if I have a gap here? I will have several reads here also, right. If I have several reads here, I can cover up this particular gap. But where did I get the clue of this region, that how do I know this contig is after this contig? How do I know that? No, I do not have an overlap. I have an insert information, right. I have an insert information. So, based on this I can identify this sequence is after this, because the read of my R1 and read of R2 is going to the other contig. So, I know this is against this, but what is there in between I do not know. That in between part will be the overlaps. The in between part, to cover this I need an overlap, but to know this is after this, my insert sequence will be telling me.
As it is random, everything is random here. So, in between the gaps, the sequence will also be sequenced using your.
Yeah, will be sequence, but you can find n still, no? Because repeats are there. There are many complex regions which cannot be sequenced with sequencers, right. So, you have repeat regions, you have other things like ambiguous regions.
Repeat regions, if it is 1 kb or 1 kb or single nucleotide repeats, then this will create problem. It's not 300.
Yeah, it will create problem, and it will skip those regions, will not sequence those regions. So, that region, to capture those regions, these insert sites helps us. Insert sites will help us, or else long sequencing will help us. So, if my long sequencing reads, one maps here, gets a significant mapping here, gets a significant overlap here, then I can say exactly this is what is the sequence here, and this can connect these two contigs together, right. This is how we connect the entire genome and get to a scaffold level of the genome. So, this is how a contigs looks, wherein the ends are not there and they are shorter in sequence, whereas scaffolds are longer, having ends, that is we can combine these two together using ends. This information comes from my insert sequence, and based on overlaps I can resolve these ends. This is the basics of assembly, how it has been done. Now, based on assembly, after what we do is that we have ends in between, we do a gap filling. How do we do a gap filling? In gap filling, we have two sets of gaps. One is physical gap, which is no information known about the adjacent context. We do not know what is. I know that this is after this, but I do not have an information between. I just have the insert sequence information, but I do not have any sequence in it. So, in that case also ends will be there. Yeah, in those cases we will have ends, and in these cases what will happen, it will break into two scaffolds. I do not know if scaffold 1 is after scaffold 2 or not, but yeah, there is some thing. So, the scaffold will be breaking there. We do not have an information in between what is there, but if we have information between, I will have ends in it and it will form a scaffold. So, this is two types of ends.
Within the scaffold, you can use an insert sequence for gap filling.
No, insert sequence.
Within the scaffold,
Yeah.
if the contigs are placed and there is a gap in between, so you can use the sequencing kind of a.
Yes, yes. So, what?
So within the scaffold, you can fill up the gap.
Yeah, within the scaffold.
Between two scaffolds, you can.
Yeah. Between two scaffolds we cannot, but we also have a super scaffold. So, what we do is, if for example, if I am using one software to do a scaffolding, it might not have got an overlap between those two scaffolds, right. But if I use some other, like I am using De Bruijn and go for an overlap one. Yeah. So, there are several software, several ways how we can connect it. So, if I use any other approach, I might get an overlap between the first scaffold and the second scaffold, then I can combine.
Essentially, you need to use more than one software to assemble all your genome sequence.
Yes. So, if you are doing for
The
job.
that depends again, if you are using one software and you have optimized it, you can use. Now, there are softwares which uses both the algorithms,
yeah.
like Masurka uses both the algorithms. So, you eliminate all your errors of assembly at least.
Yeah, okay.
Then you go for a gap filling with different softwares. So, they will again fill up. Then you have different libraries like MatePair, PacBio and all. So, those can fill up these scaffold ends in between.
Okay.
Yeah. You can use longer reads and insert sequence information which will fill up your.
So, it's all a jugglery of the best softwares and most productive softwares, as well as the jugglery of the different platforms.
Right.
Isn't it?
Right, exactly.
Single one may not give you the complete picture of it.
But if smaller genomes like
bacteria smaller genomes like bacteria and all, yes,
you
definitely.
can go with one platform
single
because
platform.
the complexity is also less.
That's
but
right.
But if you are going for fungus and all those
eukaryotic genomes, you need to have different platforms, different softwares to bring out the assembly.
Or else you need to optimize it.
That's right.
So, we have optimized on so many years. So, now we know if the sequence looks like this, the genome looks like this, so this software will work well for this.
Right.
So, we need to identify what is exactly coming out. At the first preliminary assembly, what we have the more number of ends or we have more number of contigs. What is the issue? So, that has to be identified. Based on that, we can use whichever software we want.
Let me understand, help me to understand that after making scaffolds, the regions which you are putting N to join the two scaffolds. So, you are saying that region may not have been sequenced. So, we do not have overlapping of that. Because of that, I mean, this consensus sequences are not formed for that region. So, you are saying we are joining it based on our insert sequences. So,
insert information,
insert information. So, from where we are getting that information and what
in
is
the sequencing we have an R1 and R2. In the library, I am creating 1 KB library of MatePair, right. In my MatePair 1 KB, I am just having a base of 150 and 150. Rest is all my information that something is there in between.
Yes.
There is a distance in between these two reads.
Yes.
So, assembler tries and assemble in that distance, right. So, what we give, we tell the software that this is a library with 1 KB plus minus 200 bases, right. So, it becomes 800 to 1200. So, that is what is the distance the assembler will consider, and in that distance only read 1 and read 2 will have an overlap. If it does not get a read 1 and read 2 overlap, it will not assemble. So, that is where I know ki something is there in between and will fill it with ends if I am not getting any overlaps in that gap. Yeah?
Okay.
Clear? No?
Okay, thank you.
Yeah. So, this is how contigs are created, and based on that scaffolds, and scaffolds have ends in between, and this ends are filled using gaps. This is an overall picture of how assembly looks like. We have a genome, we have small sequences of reads which are having the overlaps you can see, and has a distance, equivalent distance you will be able to see. So, they are having equivalent distance because the assembler is been instructed to find overlaps between that particular distance itself. There only it can become a accurate assembling, or else it will be a random one, and you will end up having some sequence which is jumbled up. Right. Then we have contigs and followed by scaffolds. Scaffolds you can see paired end reads. This distance is called your insert. So, this is how the paired end reads maps, the insert sequence is there, and we can end up having a draft genome. Close the gaps and have a draft genome. This was a summary of an genome assembly.
Between the scaffold, that error will be there, that you know.
Between the scaffolds,
That gap is there.
right?
That gap you are filling with that another platform like PacBio and all whatever you will use.
Yes.
Since before you told that 10% may be the error in that platform.
But
So
we will be, if we are using a hybrid approach, we will eliminate those errors using short read technology.
Again.
No, beforehand only we are eliminating. We will do the filtration beforehand of only PacBio data, eliminate those errors and then use. If I am only doing PacBio sequencing, then I do not have anything to eliminate. I will eliminate based on quality and all those things and then use it. So, I cannot differentiate whether the indels is for that particular platform or whether it was there in the sample, right. So, that is why hybrid approaches are good if you are going for complex organisms, right. So, short reads can be used to eliminate those errors of indels. So, that I will filter out in the beginning itself and then use those reads in my
Filling the gaps.
right, exactly.
This error correction is through software only, it is not manual, correct?
Nothing
Typically,
is manual software.
You use Illumina reads having
Illumina
very high
reads,
accuracy rate
right.
with this PacBio read which is having random
There
error.
are quantum and there are different softwares which can be used to eliminate these errors.
So, that means you will be correcting the PacBio reads.
Right. We do an error correction in the
correction.
PacBio reads.
That correction is done through software only.
Software only,
Okay.
using your short read technology.
For the students, that is a point actually. It's nice that she opened up the topic. You have random error in PacBio long reads, because one of the speaker highlighted it here, and Illumina has got maybe in the systematic error they say, but we combine these two, then the random error and the systematic error will be corrected by either reads. Am I right?
Yeah.
What is the difference between this systematic error and random errors?
See, random are these long reads. What happens is that the sequencing chemistry is very different. If you see sequencing chemistry, it is based on polymerase, right? What happens when you are sequencing the repeats, when the repeat comes, the polymerase slips over and cannot sequence that part. So, the read is breaking at that end and it may skip that region or may sequence something else, introducing something there, right. So, the repetitive regions, or for example, we recently did very complex fish. So, in that we were having somewhere around 85% of repeats. The output of that particular SMRT is 5 GB, but we could only get 1 GB per SMRT, because the polymerase was just clipping off and slipping down. Whether that is a theoretical explanation, but when we have the reads were not going beyond 2 KB, right. whereas it should reach up to 15 kb. So, my average read length was coming 1 kb or 2 kb max. So, that means I am having something which is just clipping off the polymerase and the reads are getting chopped off. So, there can be a chances that it is introducing something else. So, if it is introducing SNPs, indels, usually what we see in PacBio, they introduce indels, right. So, to correct these indels and all, we can use these platforms and correct those indels.
So, in this Illumina it do not slips.
Illumina, the technology is different, right?
Short
Short reads.
read.
So, it is having a adapter and the adapter is getting sequenced. So, there is a DNA template which is getting sequenced. It is not a polymerase which binds one base is not getting sequenced, right. So, the technology, the sequencing chemistry itself is very different. It is sequence by sequence chemistry in Illumina, right. So, there is shorter library, sequencing is based on DNA templates. It is a bridge amplification. You can just go through bridge amplification, you will understand what is the difference between both. Yeah.
So basically one is through sequencing. Just tell me.
Yeah. So, once we have an assembly, what we usually see is the total number of scaffolds or contigs, how much we have assembled. The lesser the number, the better it is. That means it is more closer to the chromosomal level, right. Then we see the genome assembly, which is your with gaps and without gaps. With gaps is the actual size of the genome. For example, if I am assembling 50 MB, my actual size should be between 45 to 55, right. So, that is the close by. It is not necessary always you will end up with that particular number, but usually for a good assembly we try and be close to the genome size.
So if that is 15 assemble, I can come that it's what is the exact size of what we are seeing.
But we will have a homologous genome already known, or else we can find out the genome size from any of the existing ones, or else we can also identify using in silico basis based on KMUS. So, there are softwares available to identify in silico genome size. It might not be accurate, but yes, we can identify and we can see. So, if we do not get any clue, we do identify and be close to that particular size. Yeah, so, we can use the paired end reads to identify in silically what is the size approximate. So, once we are approaching that size with gaps, we see without gaps how much. So, from here we can see how better is my assembly. If I am ending up having 50 MB and only 4 MB is my bases and rest is all ends, that means a huge difference. So, that cannot be an assembly, that cannot be a draft assembly. It should be the opposite way. It should be somewhere around 45 MB and 5 MB I am having gaps, or 10 MB I am having gaps. That is a comfortable genome assembly to be published, right. So, this is what we see. Then the average scaffold size, the greater it is, you will have maximum scaffolds like 21 kb or more, the more it is better, the assembly will be better. We also reach to 1, 2, 3 MB as well. So, that is very good assembly wherein you have a maximum size of 3 MB in de novo. Then we have N50. N50 is nothing but a cumulative value. That means 50% of your contigs will be above this size. So, based on these size, we can say whether the assembly is good or not. You can see here 241 kb is the size. That means a comfortable assembly to be published. A maximum scaffold size and minimum scaffold size and the percentages of ATGC and N. These are the parameters which has been checked to say whether the genome assembly is proper or not. So, we check all these criteria, and in any publication if you see for a genome assembly, you will find these criteria written in the paper, because these are very important criteria to be seen. These are some of the tools which are being used for genome assembly. One is SOAP de novo, Mazurka, S-Space. Mazurka is again overlapping and Dubugin both. These different softwares are based on different platforms. All softwares do not use all the platforms. So, based on which platform what has been used, that can be checked, and these softwares are also for specific region. If for example, I am doing scaffolds and contigs, if it is only Illumina platform, I can use SOAP de novo. But if I am doing a PacBio plus Illumina, I can go for Mazurka. There is EdSpace, there are n number of softwares, but this is a small list which we have listed out here. For example, once I have done my scaffolding and everything, I want to still do a super scaffolding, then with PacBio data, I can use PB Jelly, which does a scaffolding for only PacBio data. So, if I have lots of ends and I am not able to resolve it with Illumina itself or PacBio with some tool, I can jump over another tool and try it out with this, because each tool have its own chemistry, own algorithms, how they run, right. So, we can do iteratively, and then finally, we do a gap closing using gap closure, and then if you want to validate our genome assembly, we map back the reads on the genome. For example, paired end reads, I will have more number of paired end reads, because usually the genome backbone has been generated using a paired end reads. So, if I have generated the backbone with paired end reads, I map that paired end reads on the genome, and if my maximum number of reads are mapping to the genome, then what happens is that we say that genome is being assembled in proper manner. For example, I am just able to map my 10% of the reads back, rest of the reads are not mapping, that means the overlapping or whatever we have done is everything is wrong. My genome is not getting mapped. The read which we have generated, that should be at least 90 to 95% mapping back to the genome, right. After we have a genome, we do a genome annotation. Genome annotation has several steps. We can do n number of things, that is the basic, that we have a genome first. Once we have done a genome, we do a gene prediction using any in silico tool, and one of them are Augustus. We can identify the genes. For bacteria, there are like Proca, they also have an online version, wherein you can, for bacteria you can upload the genome and sit, because there are small genomes. You have online versions as well for those tools, but the tools which I am talking about, Augustus and all, they are all terminal based ones. So, you can identify the genome. So, what it does is that they have a trained model already. For example, we are working on particular genome. So, for that similar genome, a trained model will be there of that family or of that genome. For example, if you are working on Fusarium, then Fusarium already some strain will be there in that model. If not, we have to find a close related one in the listed genomes which is already been trained for Augustus model. Once we have that, then based on that we identify the ORFs, CDS or ORFs, and those will be annotated based on NR database. NCBI database we annotate and we identify those ORFs or CDS which we have identified, which gene are those. So, based on homologous search, we identified using NCBI database. So, this is where we have the gene annotation. Apart from gene annotation, we can also do geoclassification, wherein we can functionally classify molecular, cellular and biological processes. So, the gene can be classified into these geoclassifications. We can also identify different pathways where the genes get enriched, which are the different pathways available. We can do a comparative analysis, compare two genomes together and identify the differences between the genomes, sequence wise differences. We can identify the SNPs. We can do repeat identification. We can identify which type of repeats are there in the genome, what are the different classifications of the repeats and which classes of repeats are there, and how much percentage of repeats are there. Then we also have several databases for pathogen host interaction like Phibase. There are other databases also, I have few clips of it. So, those databases can be used to identify whether my genes, which are those genes which are having these interactive genes. So, I can also identify that based on blast search on these databases, right. So, these analysis can be done once we have the entire genome available with us. So, that was when we are doing higher complex genome. This is when we are doing smaller bacterial genomes. So, we have KEGG pathway, we have comparative analysis, we have phylogenetic analysis, host pathogen study, we have orthologous genes, we can identify the orthologous and paralogous genes, and we can classify them into geoclassifications. So, in annotations we can segregate them in structural and functional annotation, and we can identify the genes, we can identify the exons, introns, all those things. So, once the functional annotation is there, we can see the species distribution, because it is not necessary in NCBI it will go and hit only your species. For example, if I am doing de novo, I am doing a homologous search. So, I will have genes which are getting categorized in several genomes. So, I can identify which is the major genomes it is getting segregated in. The most top one should be your nearest one, then it is said to be that the annotation and the genome assembly is comparable and similar. Right. So, from here the top species graph we can identify that this is what is my blast hit, and this is a genome which it is matching maximum. So, my genes which are identified matches with this particular genome maximum. So, these kind of plots can be made, and we can identify whether the genome assembly is proper or not. This is the geoclassification, which classifies your various genes in different classifications, cellular, molecular and biological processes, right, and how much percentage of genes are getting segregated in these three classifications. Then we have PHI-base. This is a pathogen host interactive database, which consists of somewhere around 6,000 genes, and this gives you information of pathogenicity, virulence and effector genes. So, this is a database for these genes, and when you blast against these databases you can get those information. So, you can identify which of your genes falls in which category, whether they are virulence or whether they are pathogenic genes. So, the result after blast looks like this. So, I know this particular genome is of this class and my scaffold goes to this, right. So, this is gene number 1, gene number 2, gene number 3, gene number 4, so on, and this is what is my sequence description, this is what was gene comes. So, I can identify whether which classification it falls in. So, this is how you can identify the host pathogen interactive genes. Then we have carbohydrate active enzyme, CAS it is being called. So, in this we have information about the carbohydrate active enzyme sequences. This is also again a database. We can blast again this database and identify those information, right. In this again we have bacteria, viruses, archaea and eukaryotes. The numbers every year it has been updated. So, the numbers goes on changing. KEGG database where you have various pathways, you just have to blast again those KEGG databases. Whatever genes we have identified, we just go and blast them, identify which are the pathways these genes fall in, and these are the rough categories of the pathways which are available. So, we have a metabolism category, we have a genetic information processes, environmental information processes, cellular processes and so on, organism systems, so on. So, in these categories again we have various pathways, carbohydrate, in carbohydrate we have glycolysis and all this. So, each and every gene which falls in which pathway, that information can be found, including those basic pathways like glycolysis, Krebs cycle and all. So, all those genes which falls we can identify using KEGG database, and the pathways look like this. So, wherever you have mapping you can see in different colors, where you do not have you might be seeing in white colors. You can play around with these databases. Then we have orthologous search. As I said, we can also identify orthologous genes. How do we do it? We take the protein sequences of the ones you want to compare along with your protein sequences, and we do a ortho MCL. That is a tool which can be used for orthologous genes, and we identify which are the core genes. Core genes is nothing but this forms a cluster together, might be having similar genes together, right. So, in these 6 species we have 10,740 genes which are common. These are called as core genes. And we also have some unique species specific genes which are lying here. And we will also have some common genes between 2 of the sets, 3 of the sets and so on. So, based on your study, based on your interpretation, you can identify, and you will also come to know the clusters. For example, these many sequences we put, these many clusters it is formed, and these are the sequences which are singletons, means has not got any clustering, does not cluster with anything. So, that means it might be falling off the criteria. Based on this graph, we can identify the unique species specific genes, we can identify the core genes, and based on this we can also generate a phylogenetic tree of various species. These are the outcomes of an assembly, in silico genome assembly, and then you can also validate your assembly using RNA-seq data, because RNA-seq is said to be the coding region of the genome, right, also has your UTR region. So, mRNA we can sequence and we can map it on our genome. We should have somewhere around 95% of mapping of the RNA-seq data. That means you are covering your 95% of your coding region in your genome. That means the genome is validated enough to go for. So, that also can be done, and then we have different geoterms, geoclassification. This is what is the outcome. I will just take 5 more minutes. Right. Now, the next is your reference seq, which is there. We will just see how reference seq is done. We have already a genome sequence available. Once we have the genome sequence available, we just map the reads together. These are different reads with different sizes. In this, we do not require a hybrid assembly, nor we require a different libraries, because we already have an existing genome, right, and we map the reads. Once we map the reads, this is mainly done for studying differences or SNP. Main objective of doing this, if we want to find out SNPs, biomarkers, then we go for reference seq. So, in this, if I find a region where my reference sequence has a base and this particular read has some other base, you can see a purple line tells me that there is a different base there. The reference and this purple line has a different base. So, I will call it as SNP, right. You can also see here the base which you are able to see, those are different than the reference. If I find such cases, I will say that there is an SNP. For example, the reference has T and the sequence base has A, that means the difference is T to A. So, there is an SNP which is T to A.
I have a small question.
Yeah.
When you do resequencing, the efforts are considerably less as compared to de novo assembly.
Yes.
Correct. So, when you do this resequencing, which sequencing platform is ideally suited?
It depends again. You can also use the short read technology. For bacteria and all, short read is good enough. You can also go for PacBio, which will straight away give you a one long sequence and the entire bacteria gets sequenced off. For viruses also similar case, if you use PacBio, a straight sequence will come and you have a reference to map it and do it. So, you can also go for PacBio and short read, but it also depends upon the budget, because long sequencing reads are not cheap enough. They are costly. The short read technology, because it is a reference base, you can map it easily.
So, this is one of the important points. Most of the time, in future we will end up in resequencing only, because all genomes are already sequenced. So, you have to be very careful, and you have to be in the sense, as Pita rightly put it, the genome size will decide whether to go for short read sequencing technology or long read sequencing technology, right? Please continue.
Yeah. So, we identify SNPs based on the differences in the sequences. So, once we map the reads, right. So, what we identify is that from this end to this end of my reference, how much percentage I am covering, that is my genome coverage. In that we were seeing how much is my genome assembly, how much is the percentage of genome content in it, ATGC, that is what is my size of the genome. In this we will see if my size of genome is 50 MB, right, how much I am going to cover. After mapping, how many ends or whatever it is, how much I am able to cover across. So, that is where comes your genome coverage. So, we can, if we have chromosomal wise, we can chromosomal wise identify what is my genome coverage. If I only have one chromosome, I can identify how much is my genome coverage based on calculating the ATGC against the ends. The more number of ends, the lesser the percentage of coverage it is. That means that region I could not cover with the resequencing. Then we identify SNPs. So, this can be your scaffolds or this can be your chromosomes. We identify G to A at these locations. So, we get a file wherein it is called as VCF file, which is a raw file of SNP, wherein I can identify which base is getting changed to which base on which location. And if I want to visualize, I can visualize as we saw in the first slide. Using IGV, we can visualize individual base by base, whether exactly the tool has called the SNP or not. And these SNPs will also have a quality score with the confidence it has been called. For example, if I have 10 reads mapping at that region and only 2 reads says that it is T and rest of the reads says reference reads. For example, in this if I have G to A and only 2 reads says it is A and rest of the 8 reads says it is G, still it will call an SNP, but that will not be a true SNP. That needs to be figured out based on the read numbers which are mapping to the particular location, right. So, these are the information which we get in the file, and based on that we filter out the unwanted SNPs or false calls, right. We filter out those and only keep those which have high confidence.
So, what is the threshold limit when you say that I take only this number as a
SNP?
call,
Yeah.
SNP,
So,
and
you
when will I throw out the others?
yeah. So, again usually we take 5x, 3x. So, 3x means 3 reads should be there to call it an SNP. If we have ending up with lots of SNPs, we go with 10, 15x of coverage. So, 10 sequences should be there for that particular SNP to call a SNP is there. Like in human cases, we take 3, 5, 8x. 8 is the minimum optimum wherein we can call a SNP. For plant cases, we will end up lots of SNPs. We go with 15, 20x and we identify. And with hetero and homo, what we do is that 60-40 ratio. So, if there is 60% allelic call and 40% your reference call, I can call it as a hetero call. That both the bases are there, one allele is this, one allele is this.
So, SNP calling is based on consensus only.
Based on consensus. So, it is usually picked from the BAM file. Consensus is also picked off from the BAM file.
You got this point? Late evening, we know it's difficult to grasp things.
Late evening.
Yeah.
Yeah.
Downside.
Tired?
Looks like they're tired. Yeah, this point is very important. SNP calling is one of the hot subject nowadays. So consensus of accuracy will out of 15x, she was telling 14 of them are pointing towards A, and they will call A is a genuine SNP. Okay, try to get it.
Yeah. So, based on the read numbers which are aligning that particular region, based on that these SNPs are called. And this are different outcomes. Consensus sequence is nothing but the sample which we sequence of your genome. Yeah. And then we can annotate the SNPs based on the GFF file which will be available for the genome. So, if we are using a reference based one, we already have the annotation, we already have everything we have. What we have to just do, we have to compare the sequenced genome of ours with that particular annotation, that particular genome, and we can fetch out all the information whatever was there existing. So, this is an easier job than doing a de novo one. Yeah. Thank you, I think so. Yeah. Any questions?
I think that was a nice, back-to-back lectures on assembly. I'm sure the previous speaker made a lot of information. He made a lot of information. He told you about the genome assembly. And Arpita's lecture is only adding further points, and I'm sure it would have removed some of the confusions what we had earlier. If you have some doubts, some clarifications, you are most welcome to clarify the doubts. Kavi has got some.
Ma'am, while most of our fungal genomes may have heterozygous, what approach will be using for assembly of heterozygous genomes? Where will be ending up in a bubble? And will there be any alteration in the debridging which you spoke about?
No. See, we will use a hybrid approach. Whether it is a heterozygous genome or that will not matter much. The complexity of the genome will matter, right? So, in hybrid approach, when we are assembling, those things will get eliminated. Second thing, debridging graph only cuts the genome in smaller region. Nothing else it does, and it forms a graph.
Like in an assembly, if we are assembling a heterozygous genome, then we may end up in getting a bubble where there may be two parts rather than a single part. So
But
which one will be?
Yeah, so when we are doing a k-mer size, if I cut the k-mers in smaller regions, and if I have only, just imagine 21 basis sequences, and I find an overlap, there will be no bubbles, it will find an overlap. There are very small sequences, 21 bases, just imagine, and 21 bases into some millions. If I have 10 million reads, 21 bases into 10 million. Just imagine how many sequences are there, one after the other, each sequence is getting aligned with each other. So that point will not be validated.
Like from a single cell, there are two DNA molecules,
Yeah.
like two have different sequences. So there will be getting 50-50 probability. At this position, there may be A, and in other it may be T. So how?
By the overlapping, when I am doing with a small sequence, I will get a approximate overlap, right? Yeah. Whether both the DNA templates, when I sequence also, when I am isolating the DNA and sequencing it, I have both the DNA templates with me, right? You are saying both the cases will be there, right? 1 and 2.
Both in the sequence.
Exactly. But when I am having an overlap, will go on overlapping, right? Because I am cutting down shorter sequences. It becomes very short. 21 bases, 15 bases is very short for me to go on overlapping those sequences.
Well, when we have long reads, what is the necessity for going for k-mer-based approach where we are still increasing the complexity in case of repetitive regions? Why the OLC approach doesn't hold it?
So when we are taking longer reads, we do not go for smaller k-mers. We go with longer k-mers, right? So we will not go with 10, 15 bases. So it depends upon what is your read length as well for deciding the k-mers. For 150 bases, I cannot go with a k-mer of 121, something like that, right? Then it becomes complex. Yes, if a genome is simpler, I can go with higher number of length k-mers. But if I am going with PacBio and I cut it in 10 bases, will not make sense for me, right? So we will not go with smaller k-mers, we will go with longer k-mers.
Then one more practical question. Other than the terminal-based assemblers, is there any Windows-based?
Terminal-based ones, Windows-based, okay. So you have CLC and all which are Windows-based. You want to say GUI-based, GUI-based where you can click and do it. But still you need to understand what is the chemistry behind it, because you will have ending up with lots of options. We do have GUI-based.
No.
No, their reads will only not open. So rest of the assembly you can not think of.
And like at least for a simple like quality checking and trimming, other than these fast QC is there any GUI-based?
GUI-based filtration tools, we do have. FastQC kit is there, which is a GUI-based, what I remember. There are GUI-based tools, but then again for that also you will have to have a 16 GB or 32 GB RAM PC or computer to execute it. A bacteria can be executed on 16 GB. fungal, I'm not pretty sure how much data you are going to generate, and you will be able to do it on 16 or not.
But probably bacteria one.
Yes.
But I think
Any more questions, anybody?
no, I was just since the questions were coming up, I was just thinking that the lecture part is one thing, but probably still for you to visualize everything is still difficult. Like, I'm not too sure how much visualization you can do when she says cuts, reads, coming together, overlapping and all. So I think we should make an attempt somewhere to show the whole process to them. I don't know. If we can manage, we should.
But in the process, apart from white and black screen, they will not be able to see anything. What I spoke, they will not be able to visualize that
No,
on
for
the
that
screen.
I'm telling you, you have number of YouTube videos are there about genome sequencing technology. Number of them. Right. You have to take out time and spend a couple of hours, if not on a daily basis, at least every weekly basis. That one you have to top up. This training offered you a platform to acquire some knowledge, and how far you will be skillful, we don't know. At least the knowledge part is done. This is a deliberate. We are forcing you to sit here, right? We are forcing you to sit here up to 6:30 in a day. That means what? It's a training, a deliberate training. So only thing is you need to top it up. If you forget after 31st of December, then you will go back to the original settings. Right.
Top of the extend the validity.
Yeah.
The terms, the terminology is being used. If that you are able to understand, and if you go back and practice on it, probably slowly you will catch up.
So
That's what I feel.
you have to practice time and again.
Yeah.
Practice definitely will make you perfect in this subject. Ultimately, it is your interest. Like no bioinformatics tool can be taught in a classroom. It's very difficult. Right? People can give you some orientation. Maybe they will introduce a topic, introduce a software. Ultimately, you have to practice it. Right? Unless and until you practice it through the data, with the data what you generate from your thesis, or maybe you take some data from the database, then practice. There is a lot of opportunities, right? So I think any more questions? Anybody? Yeah, there is a question from.
You have NCBI database where n number of data is there. The raw data people submit. You can go and download and play around, especially for bacteria you can play around because they are smaller data and assemble them. And every tool whichever is available, everything has its own tutorial which manuals which you can read and do it. And there are also GUIs which are freely available. Some have some training periods like trial periods. Like your CLC has a 14 days trial period, you can try out those things and play around with it.
Yes, Mukesh.
So this is discussion session. So I'm asking now to ma'am only. So anybody, it is a question, practical question. So earlier we were doing breeding, resistance breeding, disease resistance breeding through conventional methods, like the QTL mapping and all those things we were doing. So in that, once we are having available whole genome sequences of plant and the pathogen growth, so how these whole genome sequence is helping in that breeding, resistance breeding programs. So like my question is, like we are having whole genome of pathogen and whole genome of plant. So all the AVR genes and virulence genes and the resistance gene and those things are having. For all the pathogens, we can annotate. And can we transform in one plant for all the diseases at a go? Like how it is helping? I want.
The practical approach of
difficult question to answer.
That's where the interaction of the host and the pathogen or host and the bacteria will come. It's not, if you do it individually, if pathogen separate and host separate, putting them together will not be, it will be a little difficult. But if you do this kind of a study where the interaction is taking place, then only you will probably be able to come out with some kind of a strategy. That's my feeling.
Yeah,
Anybody
so you
can
can
answer this
also do a
question?
RNA-seq wherein you can identify what are the genes falling in what, like which are the genes getting expressed in that particular host pathogen interactions and identify. You are getting to the QTLs, right? So the QTL mapping is also possible using genomes, right? You can identify the genome, go down and make a physical map when you are doing a de novo and map your QTLs on the genome. right? So you identify where exactly your genome, your QTLs are mapping, and then you can put it in your practical way. I do not know the practical aspect of it, but yes, you can identify. So these QTLs, these trade markers, these SNPs, all this can help you using these technologies. And if you want to work more closely with the host pathogen interaction, you can do RNA-seq, identify the gene expressions and which are the genes actually playing part in that particular when that it is infected. Exactly.
Anybody else can supplement this data? I think somebody is raising hand. Yeah.
Supplement.
No, you want some answer for his question. Kavi? Yes. He's having some answer.
So some in bioinformatics I did classes, so there was a genome association study. So I think
GWAS.
this data
GWAS,
is
exactly.
going to help there also. There is
GWAS.
GWAS.
genome-wide association study.
Exactly.
So maybe
So there where you can map it.
in our case, the resistance governance is mainly by monogenic or oligogenic. So we don't have to use QTL terms here. So maybe to land on gene-specific markers, this may help. And if we get gene-specific markers, then it is more easy.
What is more important is, she already put it, you need to integrate data sets. Genome data alone may not be sufficient. You have to go for transcription.
Exactly.
So you will understand which plant gene is upregulated or downregulated when the pathogen is